I decided to change the title of my talk to be a little more descriptive. But, okay, yeah, so I'm Surj. Surojit's my full name, but you can call me Surj. I'm with the graduate program in bioinformatics at Harvard. And the title of my talk is Tradict enables high-fidelity reconstruction of the eukaryotic transcriptome from 100 marker genes. So this whole project came out of two observations we're all probably familiar with in some form or another. The first is that biological networks, and in particular transcriptional networks, demonstrate this property of being scale-free, which is a fancy way of saying that there's a small number of genes that have a lot of connections, a lot of regulatory connections to many other genes, and most genes actually don't have very many regulatory connections to a lot of other genes. And then second observation, which was at least made explicit very recently, is that transcriptomes are very low dimensional objects, and in fact can be represented, at least in this paper, in terms of low dimensional polytopes. The idea being that if you're a stem cell and you're differentiating into a neuron, the fact that the cell is energetically constrained, if you're being a good neuron, it means that you're going to be doing worse as a macrophage, or doing less macrophage-like things. And so these two sort of properties led us to ask, one is first the transcriptome really of low dimension, and in particular, can we actually compress the transcriptome into a small, very small set of marker genes that we can, in practice, in prospective scenarios, just measure these marker genes and then infer the transcriptome or properties thereof as necessary. So to do this, we came up with a method called Tradict, which is a play on transcriptome predict. And the idea is we basically start with some training collection of transcriptomes, okay? And I'll talk more about that later, but let's assume that our training collection is in some way comprehensive. And what we'd like to do is basically represent the transcriptome in terms of a small subset of markers. So actually what we do first is we calculate a new expression matrix of transcriptional programs. And before I explain exactly how we do that, I'd like to just define what I mean by a transcriptional program. And it's basically a major biological pathway or biological process of the cell. And our total collection of transcriptional programs are going to represent a comprehensive list of the major pathways and processes in the cell. And our goal here will be to actually model these transcriptional programs directly, because looking at a few hundred transcriptional programs is much more interpretable than looking at 20,000 plus genes. So specifically what we do is for each transcriptional program, we look at all of its constituent genes, and these can be defined in terms of Go biological processes, as we do in this work, in terms of KEGG pathways, other types of annotations, or user-defined annotations. And we compute the first principal component of a log-like transformation on the expression of each of the constituent genes in the transcriptional program, and we obtain this new expression matrix. And then from this expression matrix, we perform a procedure borrowed from signal processing called orthogonal matching pursuit to basically represent this expression matrix in terms of a subset of genes taken from the transcriptome. And sort of loosely speaking, you can think of this as a simultaneous sort of stage-wise regression of treating each of the transcriptional program expression vectors as a target. So now that we have a collection of markers that we believe are going to be representative of the transcriptome, we need a way of building sort of a predictive model to go from these markers toward a prediction of transcriptional program expression and the expression of the remaining genes in the transcriptome. So what we've done here is, this is sort of the full generative model that we're sort of assuming of our data. And the idea is that there's some master covariance matrix between markers and the other genes in the transcriptome or transcriptional programs. And then this gives rise through a multivariate normal distribution to a collection of latent abundances. And these are sort of unobserved true abundances. And here you can see between, for example, marker 1 and 2, there's some negative covariance. And between marker 1 and sort of maybe a target gene or a transcriptional program of interest, there's some positive covariance. And it's from these latent abundances that we ultimately observe our count data or our non-negative data output, whether it's in transcripts per million or read counts. And here you can see, again, marker 1 and the gene of interest have some positive association, and it's negatively correlated to the abundances of marker 2. And so the main point of this model is it allows us to capture covariance structure in count-based or non-negative data. But of course, now we're not interested in this full model. We're interested rather in sort of the conditional model between markers and the remaining genes in the transcriptome or the transcriptional programs. And so during prediction, what Tradict will do is, well, it'll take some TPM measurements, transcripts per million measurements of your measured markers, and then using fairly straightforward Bayesian statistics, we'll calculate a maximum a posteriori estimate of the latent abundances of these markers. And then, if you recall, it's these latent abundances that are multivariate normally distributed. With these latent abundances, we can use standard Gaussian conditioning rules to obtain predicted expression measurements, predicted expression values for the transcriptional programs, and the predicted TPMs of the remaining genes in the transcriptome. Okay, so in a nutshell, that's Tradict. But remember, I told you its performance is going to be limited by sort of the training data that you have in hand. And so we turn to publicly available sources, and in particular, we turn to the sequence read archive from the NCBI. And for these two organisms, we basically downloaded as much raw sequence data as we could and processed it in an informatically consistent manner. And in total, we were, after quality filtering and expression filtering, we were able to retain over 23,000 transcriptomes across both these organisms, each comprising of over 21,000 reproducibly detectable genes. And interestingly, when we summarize these transcriptomes in terms of transcriptional programs, we only really need sort of 150 to 400 processes to really describe the transcriptome of these two organisms. And so our goal now was basically to sort of demonstrate Tradict and demonstrate it as one would use it in practice. So with our training data, what we would do is perform cross-validation experiments where we would take all experiments but one, train Tradict on all of these experiments, and then in the held out experiment, use only the expression measurements of the selected markers to try and reconstruct the rest of the transcriptome and the prediction and the expression predictions for the transcriptional programs. So we started out looking first at the expression reconstructions of transcriptional programs, and this is for Arabidopsis across the training set, again in a cross-validation setting. And you can see visually there isn't much difference between actual and predicted expression of these transcriptional programs. And if we further quantify this, if you look at how well we're able to reconstruct the expression of individual genes, on average, we can achieve sort of an average Pearson correlation coefficient of 0.64, which is good, but perhaps not great enough for practical application. But now if you look at our ability to reconstruct the expression of transcriptional programs, again, because these are linear combinations of the expression measurements of individual genes, we're actually able to reconstruct these with very high accuracy. And that was for Arabidopsis, and the story is very, very similar for mouse. So now to give you some more intuition as to exactly how these transcriptional programs or predicting the expression of these transcriptional programs can be useful. We did a few case studies, and I'll share one with you right now. And this has to do with innate immune signaling in Arabidopsis. So plants have sort of two arms of an immune system, and this salicylic acid arm, which is SA, actually inhibits bacterial infection. And bacteria have evolved proteins, effector proteins, one in particular called HOPBB1, that actually upregulates an antagonistic pathway of SA, thereby facilitating bacterial infection. So in this paper, the authors sort of did this experiment with 5 different immune signaling mutants, all varied in their ability to respond to pathogen. And then there's 3 sort of hormone treatments. So here we have a genetic perturbation. Here we have sort of an environmental perturbation. This hormone stimulates the JA pathway. This stimulates the SA pathway, and this is a buffer treatment. And we have 4 time points, and RNA-seq samples were collected at each of these time points. And so the first thing we looked at was sort of a global picture of this entire experiment. Here, samples are blocked off by the treatments, and then by time, and then individually by columns, and then each row, individually by columns, represent the genotypes. And each row represents the 150 transcriptional programs that are comprehensive of the Arabidopsis transcriptome. And again, you can see between actual and predicted expression, we do a reasonable job. And given that we're interested in particular biology here, namely the SA pathway and the JA pathway, we actually found that 2 of our transcriptional programs actually have the annotation response to JA and response to SA. And so we looked at these transcriptional programs and their expression, and we found that, firstly, that, again, predicted and actual expression line up quite well, and importantly, that they reflect known biology. So, for example, this mutant here, COI1, is a JA blind mutant because it is a JA receptor. And we can see that upon MEJA treatment, it doesn't really respond all that well. And our predictions recapitulate that finding. Whereas HOPBB1, which is hypothesized in this paper to actually enhance the JA response or sustain it, we see that in actuality that it has an initially slow response, but then it maintains a very high JA response here. And again, the prediction sort of picks up this dynamic as well. And if you look kind of across the different mutants, the ordering between predicted and actual are well preserved. If we actually go in and look at the genes that make up this transcriptional program, we see that the transcriptional program expression values are actually very reflective of the underlying gene expression signatures. So just looking at HOPBB1, for example, here we see just a few of the JA genes go on, but then at time point 5, they're much higher, and then time point 8, they're slightly more attenuated. Repeated this same analysis for salicylic acid response and response to BTH treatment. Again, you can see that the ordering between these very subtly different immune signaling mutants are preserved between actual and predicted, and that these expression trends in the transcriptional program space is well reflected by the gene expression signature. Now, of course, this was a more sort of hypothesis-driven approach where we knew what we were looking for. We knew that we were interested in the JA or SA response, but Sometimes you want to do something more hypothesis-free. And so in this case, we did a differential transcriptional program expression analysis. And we looked at the concordance of transcriptional programs called differentially expressed when using actual expression values versus the predicted values. And you see generally there's actually very high concordance between the two, suggesting that sort of just targeted RNA-seq measurements coupled with Tradict actually will give you a lot of the same biology that you would get from full transcriptome sequencing. So there's a lot I didn't actually get to share with you today in terms of further case studies, in terms of better characterizations of our training set to actually hopefully prove to you that it's a very deep and representative training set. So if you're interested, please, our paper's on bioRxiv. You can just Google Tradict bioRxiv. We have a GitHub Tradict that houses all the code. You can send me an email. Email's also in the manuscript there. And I'd just like to give you a few takeaways. The first is that I think this idea of modeling transcriptional programs or biological processes or pathways directly in a supervised manner is actually very powerful. It gives you a very comprehensive and interpretable picture of the transcriptome, and often when you're doing transcriptome-wide stuff, you're really just interested in the status of these pathways and secondarily interested in the expression of individual genes. Tradict is actually able to predict the expression of these transcriptional programs with very high accuracy, and it does a reasonable job with genes. And I think these two things taken together, actually, our hope for Tradict is that it's going to enable simultaneous screening and mechanistic investigation. And these two things are sort of separate. Normally when you do like a genetic screen, you find a bunch of hits and then you go dissect mechanism. But if you're doing transcriptome-wide screening and you see that, oh, you have several mutants that are misregulated in certain processes, this actually narrows down the set of genes you have to look at for further follow-up at the time of screening itself. So with that, I'd just like to thank a few people. The folks up here are co-authors on this work. Constantine and Paulo helped me a lot with the data download and running some of the experiments. Jeff, Vladimir, and Phil are PIs and sort of guided me through this work. I'd also really like to thank Brian Cleary and Aviv Regev at the Broad Institute for valuable discussion, and my colleague Amaro. And I'd like to just thank some of my funding resources here. Thanks, and I'll take any questions.